First, I want to thank the scientific committee for allowing me to present my work today here. I will present you my early data on the characterization of the inter-individual variability in human primary nasal cells for the evaluation of inhaled therapies. So, as you already know, the intranasal delivery represents an effective method for delivering treatment directly to the nasal mucosa. And the main advantages are a local delivery, a rapid onset, without pain for the patient, and fewer side effects than in intravenous routes. There is main indication for local delivery, but the nasal cavity provides also access to many other organs. For example, of course, the lung, the brain, thanks to the olfactory cleft, the systemic root, and the heart, thanks to the absorption of the drug through the mucosa, and allow also immunization, thanks to the presence of immune structure in the nasal cavity. All these possibilities need precise in vitro studies on human models, as we know that in vivo experimentation are very difficult to transpose to clinic. However, there is two main factors that can influence the efficacy of a drug. There is nasal anatomy, because the complexity is already known of the nasal cavity, and there is a lot of variation between individuals, depending on ethnicity, sex, or the age. And we already shown in a recent publication that this variation may impact the targeting of areas and so the efficacy of the drug. The other point is the complexity of the nasal biology. The nasal cavity contains 4 epithelia. The respiratory epithelium is the main one, with a lot of subtype of cells. The cells are able to secrete mucus, for example, or to express mucociliary activity, which can clean the nasal cavity in around 20 minutes. And we hypothesize that this variation in the human population can modify the interaction between the drugs and the epithelium. So to evaluate this, there is 2 main ways. The nasal cells first, that are cost-effective but far from the complexity of the reality, and the human primary nasal cells that can mimic the complexity of the nasal mucosa when we cultivate them in air-liquid interface. Pools of at least 10 donors are widely used in publication, but this is masking the inter-individual variability, and there is a loss of statistical power. So this is why we want to evaluate this in individual culture and to see what is these variations. So first, we want to establish a biobank. For that, we collect the cells in turbinate brushing from 42 LC donors. And in submerged condition, we amplify the cells. After 14 days, we collect the cells for cryopreservation or to amplify cells again in organoids culture. The advantage of the organoids culture is the long-term amplification of the primary cells without the genetic deviation. When organoids are mature, we broke the organoids and we cultivate the cells in inserts, in liquid-liquid expansion, then in LE culture, allowing the cell differentiation during 21 days. So first, we analyze during the LE culture the transepithelial electrical resistance and the secretion of IL-6. Both of them are the transposition of the basal cell proliferation and the epithelial structure. So here you can see the main results that we can expect as compared to the literature, with a peak at day 7 and stabilization at day 14. So this is what we expect, but here you can see high variation between donors. Then at the end of the differentiation, we want to see the main cell subtype in immunofluorescence staining. So we can see here the ciliary cells in purple, the goblet cells in yellow, and the basal cells in green. And we observe the main characteristics of the respiratory mucosa, the pseudo-stratified epithelium. And it's not shown here, but we have a lot of differences in proportion on cell subtypes depending on the donors. Again, in electron microscopy, we see the main cell subtypes, the cilia, and the creation of the tight junction. So our method successfully allows the cell differentiation in agriculture with an higher success rate than reported in the literature. So now our goal was to investigate the pathological condition in these donors. So we stimulate the cells with flagellin, which is a part of the flagella of bacteria, here Pseudomonas aeruginosa, which is responsible of the common nosocomial respiratory infections. Then after 1 day, we evaluate the secretion of the pro-inflammatory cytokines. So IL-8, IL-6, and CCL20. As you can see, we have the stimulation of the cells after 1 day with the secretion of IL-8, but for the secretion of IL-6 and CCL20, we have also a high variability, suggesting that maybe there is different pathway depending on the donor, depending on the epigenetics or the cell subtype proportions. Finally, we wanted to just validate our model for the evaluation of in-life treatments. So we use the commercially available corticoids for intranasal routes, the beclomethasone and the fluticasone, and we evaluate again the pro-inflammatory cytokine profile for different time points. So again, we see the inflammation, in apical side and basal side, thanks to the presence of IL-8. And we observe a significant reduction of the IL-8 inflammation with both corticoids. This is without affecting the epithelial barrier because there is no cell death and no variation of the tear. This method successfully established representative in vitro model taking into account the heterogeneity of the population. But it's just all the data. We have to investigate a little bit more for the other cytokine, for many other donors, and we want to characterize also other parameters like the mucociliary activity and the mucous secretion. So, in conclusion, our culture successfully established the well-differentiated respiratory mucosa, highlighting the in vitro heterogeneity present in the nasal mucosa. And we believe that the creation of a large biobank and inflammatory or infection model will allow a platform for evaluating the future of the inhaled therapies, taking into account this variability. This will, in the future, facilitate personalized medicine, host-pathogen interaction, and, of course, therapeutic studies. So thanks for your attention. Special thanks for my colleagues and for all the volunteers.